Thanks, Adele, and welcome everybody. Thank you very much for for joining us today. We have an exciting presentation set up for you, including a presentation by Jeff Clogston from Nanotechnology Characterization Laboratory in Frederick. I will start with an introduction to Spectradyne and our technology, in case you're not familiar with us already, and then hand it over to Jeff right away. And then at the end, we'll finish with some Q&A session at the end. But I'd encourage you to, if you have questions come up during the presentations, just put them in the chat at any time, and we'll collect all those and talk about them at the very end. So if you don't know Spectradyne, we make breakthrough instrumentation for accurate nanoparticle analysis. Our goal is to reshape the discovery, development, and production of modern therapeutics. So basically all the aspects of making nanomedicines and nanoparticle-based life science applications, that's where our technology gets applied. And we're here today to talk about LNPs. And in a nanomedicine context, there are really three critical quality attributes. The first one is particle size, nanoparticle size, because it directly impacts the biodistribution of the particles in the target system, as well as the dose. So, the bigger particles, they can include more payload, right? And it's also related to the stability of the particles after administration. The concentration of the particles is obviously very important because that is also the dose. It's directly the dose. It's also related to toxicity. I'm sure Jeff will talk about these things also later. Finally, it's also important to be able to quantify the amount of the payload of the active ingredient, maybe it's RNA in your LNP formulation, that's being included, because that's also the dose and related to the efficacy of the drug. So when you add all these three critical quality attributes together, we end up with the important ingredients for developing safe and effective therapeutics. So that's what these three CQAs are. They're the building blocks for safe and effective nanomedicine. And I like to illustrate that with a canonical experiment that if you're a scientist, you'll be able to relate to. It doesn't matter what you work with, actually. And that is the situation where we're comparing two different formulations. So these would be different LNP formulations, call them A and B. Typically, the way this is done is you take your two formulations, you apply them to a test system, maybe tissue culture media, maybe it's a live animal model. And then that affects some change in the system. In this case, I'm showing tissue culture and an increase, formulation B makes more fluorescent cells, right? And then there's a readout that maybe you use flow cytometry to look at the increase or difference between these two formulations in the number of fluorescent cells that they generated. Right? It's a very simple sort of generic experiment, but any scientist looking at this experiment would realize that you would never consider doing this without first controlling for the dose, the amount of material that you put on the test system to make sure that it's the same in both cases, right? If you put in 10 times as much of B, then it's natural to expect you get a bigger or a different response from that formulation than formulation A, and you have to take that into account to do good science. So the accurate concentration and size measurements are critical really for any kind of good science because it leads to controlled experiments. There are lots of ways for scientists to look at the output. Typically, this is cells based, so there's microscopy and fluorescence, flow cytometry, but there are not a lot of good tools for looking at the inputs, so quantifying the inputs. Typically, a variety of instruments are used, and they look at different aspects. Okay, so I'm showing a few examples here. In LNP formulation, dynamic light scattering is really embedded. So obviously you can have one of these instruments. Some people are using NTA, nanoparticle tracking analysis. And then for payload measurements, really the only option are these bulk assays like qubit or ribogreen assays that are indirect methods. And they're really bulk measurements for measuring the amount of encapsulated or free payload. So one thing I'll point out about these is that they're all light scattering technologies, right? So the DLS and NTA both use light scattering to look at the particle size. And that's an indirect measurement. You're not actually measuring the particle size, you're inferring it from the light that's scattered from the particles. Okay, we'll talk more about that later. And then these bulk assays are really just, they're very indirect. All they can tell you is how much of the payload got incorporated, but not how it got incorporated. So because these measurements are indirect, none of them is particularly well suited for the nanoscale. You know, nanoparticles are smaller than the wavelength of light, and so it's hard to measure them without significant assumptions about the samples. And in some cases, many cases, these techniques can lead to misleading results. And because we're talking about LNPs today, I want to spend a little bit more time talking about dynamic light scattering because it's really ubiquitous. And this will set the stage for Jeff's presentation later, in which he talks about the importance of using different techniques, orthogonal techniques, to characterize these formulations. So what I'm showing you here is an actual DLS measurement of a sample, okay? And the dashed line is what the instrument reports, as well as this Z average number. So this Z average number is the average effective diameter that scatters the light. It's like the mode of the scattering intensity distribution. So if you look at this distribution, it looks like a nice peak, very promising, and you might be excited about that. But it's important to remember that this distribution is actually, it's not a particle size distribution, it's an intensity distribution of scattered light, right? So all this really means is that the light is being scattered from this sample on average as if it was composed of 165 nanometer particles, okay? If you look at the ASTM standard for the method of DLS, it specifically says that converting this scattering intensity distribution to particle size or particle concentration distribution is, quote, ill-advised and fraught with danger. So this is pretty strong language for an ASTM standard, but it's really in there. And as a result, our take home is that we're actually not getting any concentration information from this, despite what the manufacturers may say about the DLS technology now. It's still making pretty strong assumptions about the sample in order to convert this to a concentration. So why is that? Why is it actually so weak, right? Why can't we say more about the distribution given the DLS measurement? Well, so it's saying the Z average of 165 nanometers, right? The basic answer is because multiple distributions can provide the same Z average number, okay? And let me illustrate that. So it's possible that your sample looks like this, right? You have a nice monomodal distribution of particle sizes, right? Centered at 165 nanometers, okay? It's also possible, because we're not measuring concentration, that the distribution looks like this, right? Could be a 10-fold lower concentration, you won't know that. This is still a pretty nice example. It could also be that the distribution is more complex, but it averages out to a Z average of 165 nanometers, okay? And in particular, what's missing in here, well, right. And so the only way to get the actual particle size distribution is to use a different kind of technique that's not using an average or that's not using light scattering as a measurement. Okay, so we know what's in this sample. We prepared it. It's a mixture of polystyrene beads, 50, 94, 120, and 150 nanometers. Okay, mixed together at equal concentration. And when we measure this sample on Spectradyne's technology, which I'll talk about in a minute, we can resolve all those different components. Okay. And what we see, because we know what's in the sample, right, is that the dynamic light scattering measurement really is just picking up the largest particles in the sample, because they scatter so much more strongly than other particles. Okay, so there's a lot of richness to this particle size distribution that dynamic light scattering is missing. On top of that, dynamic light scattering doesn't tell us about the payload, right? So, and neither does the bulk measurement, the ribogreen assay, where for all we know, the payload may be encapsulated in just particles that are, bigger, between 150 and 200, and nothing might be in the small ones where you want it to be. Okay, So this is just an illustration of why we need orthogonal measurements. Jeff will talk about this more. But it's a critical need because dynamic light scattering on its own is not telling you enough to be able to develop a proper therapeutic and understand what's in your sample. So this is where Spectradyne's technology comes in. This is the ARC particle analyzer. It was launched about a year ago. This is our Gen 2 technology. It delivers direct and accurate particle size measurements of LNPs, size, concentration, as well as fluorescence, so payload measurements, from 50 nanometers and up, okay? Importantly, there are no assumptions required for the accuracy of this technique. So the accuracy is independent of the refractive index, the polydispersity of the sample, or the heterogeneity of the sample. So it can handle mixtures of different kinds of materials, no problem. It's also fast and easy to use. It requires no alignment, no calibration, or cleaning of the instrument in between operation, in between samples or runs, okay? Let me tell you a little bit about how it works so that when we show you data later, you'll understand what's going on. So the magic happens inside this microfluidic cartridge. It's about a ½ inch on the side. And the way our technology works is we flow the sample through a constriction in the fluid flow path, okay? And every time a single particle goes through this constriction, we get an electrical signal, not an optical signal, this is electrical, that tells us the particle size, as well as how fast it went through the constriction. And so from that, we can get particle size and concentration because we know the sample flow rate through here. So this is our Gen 1 technology. It's been on the market since 2015. And in the form of the NCS1, we have instruments on 5 continents now, okay? In the ARC, which is our newest Gen 2 technology, what we're doing is we're exciting each particle as it goes through the sensing constriction with a laser, and we're measuring the fluorescence from the particle in up to 3 optical channels. So not only are we getting size and concentration using electrical methods, so this is a non-optical technique, we're also using the best parts of optical techniques, namely single particle fluorescence, like in a flow cytometer, to measure the other characteristics of the particle. And I'll show you how this can be used to measure payload in LNP formulation in just a sec. So the microfluidic cartridges are highly engineered. They're patented technology that include features that make them easy to use, such as an embedded filter on the upstream input of the cartridge, and very precise, consistent features. Ultimately, these engineering features aspects add up to ease of use for the end user. It allows you to measure very complex samples like tissue culture media or biofluids directly on the chip. It delivers consistent pre-calibrated cartridges, so you don't have to do any calibration. And it affords very small sample volumes for measurement, just 3 microliters required. So this ARC instrument delivers 3 critical quality attributes in a single measurement. Electrically for size and concentration, and using fluorescence for payload, and I'll show how that works. And it's fast and easy to use. You don't have to take our word for it. We won the Cyto Innovation Technology Showcase this year in Montreal. This is the Global Association for Advancement of Cytometry for being a promising new technology. And we have this technology being rapidly adopted for LNP formulation analysis. Let me start with a measurement example that just highlights how the technology works, and then I'll show you a quick one on LNPs before we hand it over to Jeff, okay? So to illustrate how the technology works, we put together a complex mixture of polystyrene beads, okay? They range just from 100 to 290 nanometers in diameter, but there are 6 different components in this mixture. They have different kinds of fluorophores on them, and they're different sizes with overlapping size distributions. When we measure this sample on the ARC, we get raw data that looks like this. So what I'm showing you here is in blue, it's the electrical signal. So this is showing single particle detection using our microfluidic resistive pulse sensing technique. And you can see this first particle that comes through also generates fluorescent signals simultaneously in all 3 of our fluorescence channels, okay? Whereas this particle is a non-fluorescent particle in the sample and does not generate any fluorescent signal simultaneously, okay? So this is how the instrument works to collect the data. Ultimately, what we do is we generate a distribution of particle concentration versus true particle diameter. Remember, we haven't inferred this from any light scattering properties of the technology. It's just directly measuring the size of the particles. We can see a few different distributions shown here. So in blue, we're showing the distribution of all the particles as detected electrically. And you can see those 100 nanometer and 200 nanometer beads sticking out prominently because they're not fluorescent and they don't show up in the fluorescent size distributions that are shown in green and yellow and red. So those distributions are the distributions of particles that are fluorescent in our FL1 to our green, yellow, and red fluorescence channels. So let's look at this in more detail because we have a lot more ways to look at this. Showing you here scatter plots. So in this first big one, this is showing a dot for every particle that's been measured, okay? On the y-axis is showing how bright the particle is in our FITSI channel, the green channel. And on the x-axis is the measured particle diameter electrically, okay? And right away, we can see clustering of different particle types. The non-fluorescent particles are below our detection threshold, the 100 and the 200 nanometer particles. And now we can start resolving the two 180 nanometer particles that have different fluorochromes. So you can see them clustered here, they're same diameter, but different fluorescence. And so we can isolate that. We could put these with a custom gate right around them and quantify just that subpopulation of interest. We can do the same thing in the two different other fluorescence channels. Okay, so the ARC is able to resolve all the components of this very complex mixture readily. You know, this measurement takes 5 minutes. Okay, so let's get into the interesting applications now and talk about LNPs. And I'll just show one example here of measuring LNP formulations on the ARC. So what we did was we took 3 LNP formulations. They're loaded with RNA to varying degrees. We stain them with a membrane permeable intercalating dye that shows up in our FITSI channel. And importantly, this is a one-step process. So you just incubate with the dye and run it on the instrument. There's no sample cleanup required. And the measurements themselves take about 5 minutes. We also ran these samples on DLS instruments. And so we have the Z average information for these samples. Okay, and that's what's shown here. So if you measure these by dynamic light scattering, this is all you get, right? It's the Z average of the measurement if you're doing it properly, right? We don't get the size distribution of particles. We don't get the concentration of either of these samples. When we run them on the ARC, and now we're going to look at just the electrical information here, so concentration versus size, we get very high resolution actual particle size distributions for each of the samples. And you can see subtle differences between them already. This blue curve has a shoulder in the distribution of larger particles in the sample. And there's a broad size range included in each of the samples. We can be quantitative. We can report, the median size concentration of all the particles on any sub-range here. These values are for the whole size distribution. So we have a lot more than just the size and concentration, though, because we measured these fluorescent samples on the ARC. So let's look at that. This plot is showing you, again, a scatter plot where each dot represents a particle being measured in the 3 formulations. We're showing the brightness. So this is the amount of payload encapsulated in each particle now on the y-axis versus true particle diameter on the x-axis. We have a limit of detection, of course, which in this case is equivalent to about 30 free floating molecules of FITSI in solution. And if we put a threshold above which we consider these to be fluorescent positive, we now report the fraction of particles that are fluorescent. We're measuring the absolute concentration of these. And so these are really the payload carrying particles that are going to be the active ingredient in these formulations. This is exactly what you're trying to measure, right? We can look at this in different ways. So now let's be quantitative about how bright the particles are. So this is showing a distribution of concentration versus the brightness of each particle. And now we can start really comparing the encapsulation efficiency. Well, it's not the right word, but how much RNA is loaded into these samples on a very subtle basis, and compare these between the 3 different formulations. So clearly, formulation C did not encapsulate a lot of RNA and has a much lower concentration of loaded particles than the other 2 formulations. Finally, we can apply a gate to select just the payload carrying particles, the fluorescent positive ones. And that's what's shown here. And look at the size distribution now of those payload carrying particles. So when we look at just those payload carrying particles, now we see significant differences between the samples, where the formulation A contains payload carrying particles that have a significantly larger median size than the other formulations, and the concentration of payload carrying particles in formulation C is quite dramatically lower than the other 2 formulations. So these inform the formulation process and are important for why you want to use the ARC to measure your formulations, your LNP formulations. Okay, so just to summarize real quick, the ARC makes direct and accurate measurements of size, concentration, and payload, those 3 CQAs we talked about, without any assumptions about the content of the sample or the heterogeneity or the polydispersity. And it's fast and easy to use. Remember, there's no calibration, alignment, or cleaning required for these systems when they're deployed. Okay, so thank you for that. And now let's hand it over to Jeff, who will present the importance of using orthogonal analytical techniques for characterizing LNPs and nanomedicines, and leverage his real experience at the Nanotechnology Characterization Laboratory in Frederick. And I will stop sharing here, and Jeff, you can take over.
Okay, thank you, Jean-Luc, for the introduction. And while I load up my slides, I want to thank Jean-Luc, as well as the organizers, for this opportunity to speak today, to share my experience at NCL, and also to go through some of the results we have with the collaboration with Spectradyne. Let's move this out of the way. Okay. Okay, you can see my slides, right? Okay. All right, so before I go into the bulk of my talk, I want to discuss first what the Nanotechnology Characterization Lab, or NCL, stands for. So it was established back in 2004 as a collaboration between NCI, NIST, and FDA with 4 primary objectives. The first one is obviously characterizing nanoparticles using standardized methods, to conduct structure-activity relationships, or SAR studies, to facilitate regulatory review of nanotech constructs, and finally to engage in educational and knowledge-sharing efforts, which is what we're doing today. We do work with the extramural nanotech community. They apply to us via a white paper process, and once accepted, they go through an assay cascade, which consists of physical chemical characterization, immunology, and pharmacology and toxicology. We work with FDA and NIST throughout this process, so we receive guidance from them. And we also have a small formulation group within NCL as well to help reformulate any drugs that you may be working with. The output to this is that we develop standards and SOPs, and we work with ASTM, USP, and ISO. And we have a lot of standards being or in process with them right now. We do preclinical development, clinical translation. We help with any regulatory concerns, and finally help with commercialization. We've been here for over 19 years now. And really, the main take home from this slide is that we help accelerate the translation of promising nanotech drugs and diagnostics. More information can be found on our website. And I should also make mention that this assay cascade is also a free service. So a lot of characterization done for free. All one needs to do is apply to us through the white paper process, which can be found on our website. We have 3 sections, as I mentioned, our assay cascade. We have PCC, which I head up. We have in vitro and in vivo immunology, which Dr. Marina Dobroskaya leads up. And then finally, Dr. Steve Stern heads up the in vitro and in vivo pharmacology and toxicology sections. What's unique here is that we have all 3 of these sections contained under one roof. So we can actually link all these three different attributes to biological outcomes. And I think that's important because not a lot of labs have this kind of capability. It's usually have one or the other, not all three. We worked with over 500 different nanomaterials. On the left-hand side is a cartoon representation of the different platforms we worked with. These are the more common ones. And we worked with a wide range of APIs, so your therapeutic agents. These can be your small molecule chemotherapeutics, your neoantigens, as well as your therapeutic nucleic acids. We have expertise in characterizing targeting ligands, as well as surface coatings. And really what we do is we work with you, the consumer, to develop a research plan tailored to your formulation, which considers your intended clinical indication, dose, and route of administration. Finally, some just metrics on here. We've worked with 23 collaborators in clinical trials, a handful of IND, NDA submissions that we helped out with, several market authorizations, and two that I want to make point here is Salvation, the COVID prevention OTC spray, as well as we worked with Addis Bioscience NAV sirolimus for cancer. With that, I want to also thank my group here. So this is a picture of all the people within our NCL group, who I thank them all for all their work and their support throughout the characterization. They actually do all the work for us and make this happen. So with that, now let me switch over to the bulk of my talk. But before that, I want to make sure that we're all on the same page with critical quality attributes. I know Jean-Luc went through this already, and I think most of us here are familiar with this, but just to kind of refresh our mind, the CQA is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit range or distribution to ensure the desired product quality. So what does that mean? These must be clearly defined along with all potential risks due to changes. This can be anything from nanoparticle attributes. It can be the drug components, excipients. You have to consider the intended use and route of administration. And what makes this challenging is that it's actually unique to every formulation. I listed here several, on the left-hand side, several quality attributes that pertain to nanomedicines. I know Jean-Luc covered a couple of these already, namely size, particle concentration, as well as payload, and those would fall under size and composition. And it's challenging here because we need to identify which of these greatly influence the biocompatibility, or the PK, the biodistribution, clearance, toxicity, efficacy, or bioaccumulation of your nanomedicine. And this is very challenging because not only must we define which one it is, we need to measure it as well. Within our lab at NCL, I listed here all the different types of instrumentation that we have. You can see we have a nice range of instruments here. On the left-hand side, for the most part, these are all our sizing instruments. So we have DLS, small, laser diffraction, electron microscopy, AFM, as well as resistive pulse sensing. We have a whole suite of chromatographic separations, as well as mass specs, as well as spectroscopy. And then we have a couple of these few, I would call them outside instruments. These are CHNOS elemental analysis, your thermal analysis, which is TGA, DSC, and finally a quartz crystal microbalance. What I want to do today, though, is focus on the sizing aspect and how important it is to measure using different methods, orthogonal techniques. And here at NCO, we have identified the CQAs. You know, for over the last 17 years, we identified the CQAs and the metrology needed to support the most common nanoparticle platforms. This involves liposomes, polymeric, and colloidal metal nanoparticles. On my next slide here is basically a characterization guide that we do here. So I've listed here the 8 critical quality attributes that we look at. For the top row, I list here the techniques that one can use to assess these. On the bottom, it's more of things to consider. What I want to point out here is we want to focus on size and size distribution today. And I'll give you examples of how we use each of these techniques to characterize nanomedicines. So I don't have to tell everyone here, but I think most people are familiar with dynamic light scattering. Jean-Luc did a good study on that already in his earlier talk. So all of us, I think, have done DLS, have made measurements on DLS, probably still do DLS in the lab. So this is an example. We're looking at a lipid protein nanovesicle, and we measure the DLS for it, and the intensity distribution plot is shown here. As you can see, there's a main fraction or main size population below 100 nanometers, but we're starting to see something over a micron. For those of you who do DLS, you know that DLS is not suitable for over a micron size particles. So what we do is resort to a different technique, laser diffraction in this case, which is better suited to looking at larger particles. And indeed, we do see this larger size population, along with that smaller size population that we see in the batch mode DLS measurements. We also took analysis a step further by running asymmetric flow field flow fractionation, or AF4. I'll discuss this a little later on in my presentation. But basically, this is a separation based off of size. And with inline mols and DLS, we can look at the sizing of a fractionated sample. So in this case, we're seeing the nanoparticles, which we saw from the DLS and the laser diffraction. We see these larger size lipid aggregates, which we kind of see in our laser diffraction. But now we're seeing free protein, which we did not see in the other two techniques. So again, it's very important to use orthogonal methods if possible when characterizing your particle for size. For those of you working with lipid nanoparticles or liposomes, cryo-TM would be your second technique that you use, with the DLS being your first. This is just an example of how we use cryo-TM in our lab to look at liposomes. You can measure, obviously, the diameter, and from that, you can get particle volume. You can look at bilayer thickness and things like that. You can also look at the morphology of the drug as well as the liposome in this cryo micrograph. However, TM does have its limitation. Besides being, time consuming, you need to sit there and count every particle. It might not always be the best way. And here is an example of the limitation of TEM. And this comes way of a pegylated metal oxide nanoparticle. We were asked to look at 3 different lots and to see if TM can be used to assess batch-to-batch consistency. I show you here lot 1, 2, and 3, the TM micrographs, and then you can measure the average diameter. They look the same, correct? You really can't tell or discern any differences between these 3 lots. However, if we use AF4, which can separate based off of size and in line with mols, multi-angle light scattering, you can look at the size distribution across the peak shown here. And you can see that one of the lot is very different from the other 2. So in this case, TEM wasn't able to discern any differences, whereas AF4 mols was. Again, highlighting the importance of orthogonal methods for assessing size. So I mentioned AF4, this is up and coming. We use this a lot in our lab. I showed here just a side schematic of an AF4 channel. So let me kind of walk you through this. We have basically a channel thickness that's defined by the spacer. We have a bottom semi-permeable membrane, typically a 10K regenerated cellulose cutoff membrane, and then we have a channel flow. The separation or driving force for this is a cross flow, which basically pins down the larger particles and allows the smaller particles to elute first. So that's the order in this case. And there's no stationary phase, like I mentioned. It's a membrane only, and we can change the membrane, the spacer thickness, and cross and channel flows to affect the separation. On the right-hand side here is just what it looks like set up in our lab. Let me just, there we go. So here's the AF4 channel here, and then again, I mentioned we have downstream detectors, so UV-Vis, mols, RI, and DLS. So our typical setup is to use AF4, which separates the nanoparticle. Then we have inline DLS to measure the hydrodynamic size, as well as inline mols, so look at the radius of gyration. And then you can always collect fractions coming off of this to do offline analysis. In this case, we can look at drug concentration. So I want to just give you some examples of the utility of AF4 because we use this a lot. This is an example where we have 30 nanometer colloidal gold with different peg lengths, namely a 2, 5, 10, and 20K. If we were to mix these 4 samples together and run a batch mode DLS, that trace is shown here in the dashed line. Obviously, batch mode DLS cannot resolve these 4 species. It's a limitation of a batch mode DLS measurement. And for reference, I show you the 4 here. So clearly, it's obvious that DLS fails in this case. They're trying to resolve these 4 species. However, if we hook this up with AF4, we can separate based off the size. And then with inline mols and DLS, we can now start to see the different size populations within this mixture of samples. So I'll just give you some more examples. So to show that not only does it work for colloidal gold, but it works for other types of platforms. For example, is a drug-loaded liposome. If we do a batch mode, the intensity distribution is shown here. It's polydispersed. If we run this through AF4, DLS, we can see the sizing of these different fractions. But you can also do offline analysis to look at drug quantitation. Drug quantitation is important, is one of the CQAs, as Jean-Luc pointed out in his presentation. This allows us to do that as well by collecting these fractions and then doing post-analysis. We can also look at drug-loaded micelles. I kind of like this example better because if you look at the batch mode DLS, it looks very polydispersed. And AF4, in terms of resolving power, is very good in resolving these two size populations. And not to leave out everyone, we also work with LNPs as well. So we can use AF4 to separate LNPs. In this case, we show the AF4 DLS trace down here, and we collect fractions coming off of the AF4, and we do post-analysis, namely to look at lipid composition as well as mRNA distribution as a function of size. And we can also use this to look at protein binding assessment. So we can incubate the samples with and without human plasma, run AF4. AF4 can separate the free unbound proteins. And we can then look at what happens before and after incubation with human plasma and calculate the shape factor to qualitatively assess protein binding. So in this case, there is none because these factors are roughly the same. We can also collect fractions, as shown up here, and do offline analysis to look at particle per mole concentration as well as zeta potential. So it's a very useful technique. However, everything I described so far are ensemble methods that are basically they measure the average or bulk properties, which is fine. But for example, DLS cannot give the overall size of a sample, but it can tell you how many particles of a certain size. If we're looking at total API or active pharmaceutical ingredient, let's say a drug, for example, it gives us the overall drug concentration, but it does not measure how the API is distributed over the size range. We can kind of do this using AF4 and collecting peaks, right? However, it cannot tell me how many liposomes have API or how many are empty. The size is the same for both populations. And finally, if they are loaded with API, to what extent is it loaded? So these are kind of questions I always have, and I ask when I approach new technologies to see if they can address these issues. And I think where the field is moving towards is that we need more advanced analytical techniques because the formulations are becoming more and more complex. And I think the field kind of needs to move into a per particle basis characterization. So we need methodology for that. So the first step is to look at particle size and particle concentration. That is where you're starting to count individual particles. And in our lab, we have two instruments that are based off of light scattering. As Jean-Luc pointed out, these are indirect measurements. We have two instruments based off of resistive pulse sensing, which are now looking at individual particles. And finally, we have a single particle ICP-MS, but this is only relevant to metal-containing nanoparticles. So we have many different ways to look at particle concentration. And I think this is important, again, to highlight the use of orthogonal methods. And really, in this case, there are no reference materials for particle concentration as of yet. So it's important, if you can, to use several of these to measure particle concentration and kind of compare and contrast. Typically, in our lab, we'll use at least two of these when assessing particle concentration. So an example to show you that it actually does work, we're using the NCS1, so Spectradyne's first instrument that we purchased many, many, many years ago. And we use this to look at liposomes, namely Doxil and Sunpharma, which are two doxorubicin-loaded liposomes, as well as Onivide, which is an irinotecan liposomal formulation. And from this, we can see the results here. We can measure the size as well as the sample concentration. So this is a very good first step in starting to look at individual particles. Now, of course, we need to see what more can it do. And again, like I mentioned, for emerging technologies, I always ask these questions. So I want to know the resolving power in terms of a mixture of different size populations. Can I look at, non-API loaded versus API loaded? Can you measure the amount and the load distribution? Can you look at free API? And then finally, can you measure the release of API in plasma? So if you can kind of find an instrument that can do all of this, that'd be your be-all, end-all instrument. And I think it answers a lot of the CQAs needed if you can do this in one instrument. So for this, we actually approached Spectradyne with their new technology, the ARC instrument. I'm not going to go through this because Jean-Luc already went through this, but we did approach them with the project with the same questions in mind. And in this case, we had LNPs made with the different mRNA encapsulation efficiency. So we asked them basically, can you tell me what the payload is in each of these formulations? And so we provided to them mRNA LNPs, and these were prepared and characterized by a formulation scientist at NCO, Dr. Avanti Karatni. She did some basic initial characterization. This includes DLS, which pretty much everyone does, as well as looking at free total encapsulated RNA using a ribogreen assay. So we have 3 samples: this high efficiency encapsulated RNA, which is designated here by 97% encapsulated. We have a low encapsulation efficient sample, which is half, 50%, and then we have our empty LNPs to serve as a control. So these samples were provided to Spectradyne, and they analyzed these samples on their new ARC instrument. So in addition to running these 3 samples, they also did a dilution series where they titrate the high EE LNPs into the empty LNPs. These samples were all staying with Cyber Gold for 30 minutes to stay in the RNA and to help visualize the fluorescence. We looked at 2 different cartridge types, a C400 and C900. Each of these contain a different size range. I will show you the results for the C900, but we get similar results for the C400 as well. In terms of fluorescence, we're exciting 488, and we're following at these 3 channels with the wavelengths designated here. So let me walk you through the data now. So this is the first step, was to look at particle concentration. So here's a concentration as a function of particle diameter for all 3 samples. As you can see, they're very similar in terms of stock concentration. We then looked at the individual samples in terms of fluorescence. So the first sample we looked at was the empty LNPs. And from the results here, you can see that no fluorescence signal was detected from those staying with Cyber Gold, which is a good thing. We expect nothing here. So this is a good control. And then we start looking at the other 2 samples. In this case, this is a low encapsulated efficiency LNP fluorescence. So we're starting to see some particles fluoresce. Roughly 21% are brighter. And then obviously with the high encapsulated efficient LNPs, this number goes up 30%. So it's a very good step. The data, if you put them over together, you can see that the empties are truly empty. And those, as you increase the encapsulation efficiency, the fluorescence goes up. So it's a very good technique, or a very first good step in starting to look at individual particles, not only in terms of particle size and concentration, but also to start looking at payload. We also did a dilution series whereby we titrated the high encapsulation efficiency LNPs into the empty level LNPs. And then based off of that, we can basically look at what we expect versus what we measured, and there's good correlation between the 2, basically suggesting very good robustness of the instrument and the technology. And then some summary here, we saw the same concentration in all 3 samples. Mainly the same thing we're seeing here is that the high had more mRNAs and the empty had none, which is what we expected to see. Obviously, there's more work to be done in this field, and we're actually collaborating with Spectradyne using their new ARC instrument. In this case, what we're doing here, again, we're using mRNAs with LNPs, but I want to kind of compare and contrast how the ARC works against AF4. Like I mentioned, AF4 is something we use a lot in our lab. It's a very good technology in terms of separating, but it's a lot of work that needs to be done for a given run. You need to have the expertise to optimize the methodology for AF4, then you have to collect fractions and do offline analysis. So again, having one instrument that can do all of that for you is ideal. So we want to kind of pit the ARC versus the AF4, and we're looking at the host sample versus a fractionated sample from AF4 and comparing the results to see if the ARC can give us the same results from the fractionated samples from the AF4. And we're going to include characterizations such as DLS, cryo-TM, and concentration, both by MRPS as well as light scattering techniques to kind of compare and contrast. So with that, I will stop. And I thank everyone for your attention. And I guess we can now answer any questions that you may have.
Thank you very much, Jeff. That was a very thorough presentation. And boy, you guys use a lot of different techniques over there. It's great. It's a powerful resource for pharmaceutical industry. You said you can just apply with a white paper, right, to have your formulations tested. Is that right?
That's correct. So there's more to just applying them, and you can check our website for details. Basically, you have to have an application or indication for cancer therapeutics. So you have to show demonstrated efficacy in a biological system. Obviously, have physical chemical characterization, things like that. But yeah, it's an open, free process that if you're accepted, all of this is free for you.
Oh, that's cool.
Mm-hmm.
All right, thank you. Let's take some questions from the chat and from anybody who's in attendance that wants to raise your hand. Go ahead and send in the questions. So I'll share my Q&A screen here. Can you stop sharing, Jeff, and then I will.
Okay, there we go.
Mine. Thank you. Awesome.
Mm-hmm.
Great. Okay, cool. So moving on to the Q&A portion. So please either raise your hand, we will come find you, or put your questions in the chat. One of the questions was from Dario was, how is the flow rate measured in the ARC, and what's the total volume sample analyzed? So when we talked about the electrical signal generated from each particle, one of the things we measure is how big the particle is, right? And the other thing is how fast it goes through the constriction. And so that the transit time of the particle is what tells us the sample rate of the volume flow rate through the constriction. And that's how we measure. So effectively, when every time we measure a particle, so this happens, many thousands of times in a measurement, we are measuring the flow rate at the same time, and the transit time is calibrated at the as a parameter for the cartridge that's factory calibrated. So it's not something you have to do. Hopefully that answers your question. And the second part was, what is the total volume of sample analyzed? The total volume of sample analyzed is small. It's a small fraction of the 3 microliters that you put in. And some people initially expressed reservation about that. How can you measure the concentration on such a tiny volume and extrapolate to a larger volume? But the answer is really in the statistics. So if the sample is homogeneous, which we have to assume for any measurement technique, then the uncertainty in the measurement is only dependent on the number of particles that are detected. So assuming you count enough particles, then that defines the certainty of the measurement. So hopefully that answers your question. Fabio is asking how many measurements can be made with the same microfluidic cartridge. The answer is that the cartridges are designed for single use, okay? And that affords all the advantages, that delivers all the advantages that we talked about. So our customers love them because they don't have to do any cleaning, there's no alignment, there's no calibration required. You just prep your one sample while the previous one is measuring, and then switch over to the new cartridge. Fabio, another question. How large are actually the pores of these different cartridges? Okay, so, and the next question, the follow-on to that is, how do they clog, right? So let me show you that one, answer that question. So with the TRPS, this is a different implementation of the resistive pulse sensing technique. Those instruments, they're made by a different company. They do suffer a lot from clogging. Part of Spectradyne's innovation is in the microfluidics, where we have embedded an input filter input to the cartridge. And so what that does is, and it's not a membrane filter like you would normally think of. It's really just a series of small channels, a network of small channels through which the sample must pass. And so any larger particles than the constriction get caught in this filter and don't reach the constriction. So that allows our users to run very complex samples like plasma to do real life PK, PD studies of nanomedicines directly in the plasma, tissue culture media, you know, cerebral spinal fluid, just straight on the cartridge. Okay. Let's see what else we have for questions. There's a question about the cost of the cartridge. They're $12 each, and remember, there's no cleaning required, so most people love that deal. Essentially, it saves a lot of time. And follow-up question to that is there a possibility to measure your samples? And definitely the answer is yes. So we routinely take samples as demonstration of our technology from customer prospects and people that are interested in the technology. So reach out to us, and we can talk about doing that for you, for sure. Thank you. We'll follow up after the meeting too. So let's see what else. Any other questions at this point? Oh yeah, there's another question about the dynamic range of the different cartridge sizes. So I will show you an example of that actually, and how the data stitches together. So because these cartridges are measuring the concentration in real terms, so it's particles per mil, right? And the particle diameter in real terms, the data just stitched together, okay? And so this is what I'm showing you here. This is a measurement of an LNP sample, actually, that it was just on the NCS1, but it demonstrates how the measurements from different size cartridges just stitched together. You know, we're measuring different portions of the same particle size distribution. And so this measurement shows the measurement of a single sample in 3 different cartridge sizes, right, where we're sampling different size ranges. And you'll see the concentration of the sample varies by like 9 orders of magnitude within this sample in concentration. And we're measuring from 65 nanometers up to, you know, 6 microns essentially here. So many orders of magnitude in diameter too. So it's quite a broad size distribution, or broad dynamic range of concentration and size. Hopefully that answers your question. Is it possible to increase the volume of sample? Yep, we can talk about that. And so what I suggest, Fabio, is we set up a call to discuss your questions in more detail, and we can answer them together in real time. So we will follow up with you after the meeting. Okay. Any additional questions from the audience? Okay, great. Well, then I will end it off here. This will be the end of the meeting then. So first, I just want to say thank you very much to Jeff for joining us today, and Dr. Clogston, for presenting your all to do about the NCL. It's a really powerful facility, and I would encourage everyone to go and check it out, how it can be of use to you. And thank you to all of our attendees, and we'll see you soon. Please reach out to us if you'd like to learn more. So this will conclude our webinar. Thank you very much.
Bye, everyone.
Bye.